In this video, we will discuss about, or we will continue our discussion of this RNA binds to glycan. In the previous video, we ended at a point that it seems like high molecular weight RNA binds to the glycan, as it can be seen in the RNA blotting. So So in the panel A, what they have done is they have tried to enrich the proteins that are polyadenated, as it is speculated that uh, high molecular weight protein needs poly A tail in order to get stabilized. So they enrich the proteins through this poly A enrichment process, and then try to see whether they are able to uh, find out any high molecular weight proteins or not. But contrary to their belief, they did not see any high molecular weight protein, even though they inject three times the RNA samples compared to the control. So from this experiment, uh, come to a they starts to think that it's not the high molecular weight RNA that is attached. However, the restriction of the flow of this RNA could be because of the glycans that is attached to the RNA. So, in order to check this further, what they did is they used another approach, that is precipitate RNA based on its length, differential differential separation of RNA. And in this, when they did their experiment, this experiment is about fractional separation of RNA based on their length. So they separate the high molecular weight RNA and the low molecular weight RNA, and then run the RNA blotting, and uh, contrary to their previous assumption, they found that most of the RNAs are less than 200 nucleotides. So these are the small RNA that is bind to the glycan, not the big RNA. So later on, what they did is they checked this small molecular weight RNA through this gradient, through the RNA blotting in the sucrose gradient from 15% to 30%, and we can see that in the uh, we can see the small RNA can be is are visible. Small RNA are visible in the sucrose gradient in the initial percentage of the sucrose gradient, that is around near close to 15 to 20%, and we can see that in the blotting. So this shows that the small RNA are present. Small RNAs are present, or small RNAs are could be could small RNA could bind to the glycans that is detected by this experiment, that is designed based on click chemistry. So then they try to do RNA-seq of this small RNA, and when they did this experiment, they are able to find out that some of the RNAs are upregulated and those RNAs plays an important role in the cellular functioning. So one of the RNAs that they pick is the Y5. In order to check whether this RNA sequencing is correct or not, they did another experiment in which they prepare a HeLa cell lines, in which they knocked out this Y5, and then repeat the same experiment to uh, bind the RNA to you can say incubate the cells with their probe that can detect this glycans bind to the RNA. And in this experiment, what they have find out is that there is a decrease in the amount of signal in the wild type compared to the knockout. So this thing, then they quantify this thing through in which we can see that the p-value is less than 0.05, that is 0.033. So this further substantiate the points that the point that the RNA-seq data that they have occupied is correct and there is a decrease in the 30% signal in the knockout RN5 knockout sample compared to the wild type. So this figure made a point that small RNA molecules bind to the glycan and not the big RNA molecules. So I hope you have understood the main goal of this figure or the purpose of this figure. If you need to go through the whole story in a single attempt, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.